Okay, guys, so we're just going to dive right in because I know you're all tired from the exam, right? So I know you all would just maybe like to go home and sleep if you can. So, folks, we're going to be using our handout that we passed out last time, the Chapter 9, Chapter 11, Chapter 13 handout. And again, this is based on Dr. Holland's amazing lab notes. I just added some steps for the protocols, the procedures we're going to do, but we're going to be following this. And unfortunately, guys, all my copies are gone, so I apologize. If you don't have one, maybe see if your partner and you can look on together. And the file, you guys, is on Canvas, okay? So again, folks, let me just give you the kind of like the heads up here. So we have to be in full PPE today, so gloves, goggles, lab coat, and lab shoes. If you have long hair, make sure you tie your hair back, and I've got new hair ties if you need them. You guys, each of you will work individually today. You can ask questions of one another, but the hands-on, it has to be you alone. And the reason for that is the very last big experiment we do is called the unknowns, where you'll be given 2 unknown bacteria, and you're going to use all of your skills to identify the unknown bacteria down to the species level. And you cannot ask for help. Nobody can help you. So we want you to start practicing doing these procedures by yourself without the help of a colleague. You can ask your colleague questions, but we want you physically doing everything. All right? You guys, we're going to be turning on our Bacti incinerators because we're going to be doing aseptic or sterile transfers. So if you can, you guys, just try to minimize the flammable objects on your bench, right? Have as little paper on your bench as possible. And again, folks, you're going to need your test tube racks today, and you'll also need an inoculating loop and inoculating needle. And I know in the small lockers you probably only have one inoculating needle, but the procedure you're going to use the needle with, it will go really quickly. So you can just share your needles, okay? All right, you guys, we're going to start with our handout, right? So first of all, you guys, Chapter 9, Unit 9 is on motility, and we're going to focus on bacteria, so bacterial motility. So what is motility? The ability to move, right? And maybe from lecture, you guys, it was a while ago, you recall that when we were talking about the parts of bacteria, we said the flagella were used for motility, right? It's the most common means that bacteria use to move around their environment. So you guys, as evolutionary biologists, why is motility a survival advantage? Why do you think being able to move is a survival advantage for bacteria? Oh, yeah, that was good, you guys. I heard move away from maybe toxins, right? And why would it be an advantage if a bacterium can move if all the food or nutrients are gone somewhere? Yeah, it could move somewhere, right, where maybe there's a better supply of nutrients, right? So it's definitely a survival advantage to be able to move. Okay, so folks, on the cartoon here, this is showing you the difference in the structure of gram-negative bacterial flagella and gram-positive bacterial flagella. You do not need to know the difference for our lab exam too, but I do want you to know the 3 components of a bacterial flagella. Okay, so you guys, the 3 parts are the basal body, which anchors the flagellum in the cytoplasmic membrane and cell wall, the hook, and then the long thread-like or hair-like structure is called the filament. Okay, now I do also want you guys to know what are flagella made of. Any thoughts, you guys? And guessing is good. Remember, you guys, you learn better if you guess. Protein, awesome, right. So the bacterial flagella are made out of protein, and we'll see that's going to be really important because those proteins will help trigger antibody production, and we'll talk about that in just a little bit. Okay, so folks, you might remember from lecture we said that the bacterial flagella, they rotate like a boat's propeller. And when the flagella are rotating in one direction, it sends the bacterium on a straight pathway called a run. So I'm doing a run right now, you guys. And then the bacterium will stop and reverse the rotation of the flagellum, and that will cause the bacterium almost like do somersaults in space. That's called a tumble. And then it'll reverse flagellar rotation and set off on another run. So when we're watching bacteria, say if we did a wet mount, you guys, we'd see them doing a series of runs and tumbles. And it looks like it's kind of erratic, but if we were patient and we were to watch them over time, we would see they're making progress in a given direction, right? So we're like, well, what's guiding them, right? So there's different environmental clues that can guide them. And one kind of guidance is chemical gradients in their environment. So the bacteria may have chemoreceptors, receptors for specific chemicals on the cytoplasmic membrane. So when the chemical binds to the receptor, it will trigger the events that determine, does the flagellum rotate one way or the other. Okay? So you guys, let's say the bacterium is moving along a nutrient concentration gradient, right? So do you think the bacterium wants to move from low nutrients to high, or from high nutrients to low? From low to high, right? It wants to find more food. So that's called positive chemotaxis, when you're moving up the concentration gradient. But in contrast, you guys, what if the bacterium encounters a poison or a toxin in the environment? Does it want to move from low poison to high poison, or from high poison to low poison? From high poison to low poison, right? So down the concentration gradient. That's called negative chemotaxis. Good. You guys, what if you were a photosynthetic bacterium that had flagella? What's important for you to detect in your environment? Light, yeah. So what would we call movement along a light gradient? Phototaxis. Good job. Excellent, you guys. So on your handout, folks, we are not going to talk about magnetotaxis. That's movement along a magnetic gradient, I guess. We're not going to talk about that. So, and I won't ask that on the lab exam. Good job, you guys. Excuse me. Okay, you guys, we're going to turn to page 2. And up there, this is a really good lab exam type question, you guys. We want to know the difference between true motility and Brownian motion. So Brownian motion, you guys, if we looked at a wet mount and let's say we had any cell there or a particle, Brownian motion, it appears like the little cells are just jiggling in motion. And it's from being bombarded by water molecules or other substances in that liquid environment. So Brownian motion, you guys, it's not true motility. You know, the cells are just sitting there getting pushed around by water or by other substances in the environment. So is Brownian motion true motility? No, it's not. Good. For sure, folks, on the lab exam, we usually have a station with either photomicrographs or diagrams of the different flagellar arrangements on bacteria, right? So sometimes flagellar arrangement is a hint or a clue as to the identity of an unknown bacterium. So you guys, not on the handout is a term that I would like you to know, and the term describes a bacterium which lacks a flagellum. And the term is atrichous. Atrichous. Without, it literally means, you guys, without a hair. Trichous refers to the hair, the flagellar filament. So atrichous means no flagella. Would you predict that an atrichous bacterium would be motile? No. Probably not, right? There are other kind of really interesting different ways bacteria can move. But for us, you guys, we're going to say if the bacterium lacks a flagellum, if it's atrichous, we're going to guess it's probably non-motile, right? It can't move, right? Okay, but now, folks, we're going to talk about what if the bacteria do have flagella. How do we describe the different arrangements? So you guys, let's just walk down the cartoon here. So you guys, what do we call it if the bacterium has a single one flagellum? What's the prefix we use for one? Mono, good. So you guys, so if a bacterium has one flagellum, what's the arrangement called? Monotrichous, awesome. And the next one, you guys, if bacteria have a tuft, a bundle of flagella at one end, what do we call it? Lophotrichous, lopho means a bundle, good. Folks, what if we have one or more than one flagellum at each end of a bacterium? What do we call that? Amphitrichous, right, for both ends, good. And my favorite one, you guys, is the one, the last one there. It's when we have flagella covering the surface of the bacterium. What do we call that? Peritrichous. Peritrichous. Do you think that bacteria that have peritrichous flagella are going to be really good swimmers? Really, really motile. Yeah, they're going to zoom across the surface of your agar plates. Well, probably not zooming, but pretty doggone fast. Good. Okay, folks. So let's see. What else do we have to do here? Okay, so another cool way, you guys, that we can identify bacteria that's related to their flagella. Remember how we said flagella are proteins, right? Okay, so it turns out, folks, that the flagellar proteins are what we call really good antigens. And antigens are substances which trigger what? Folks in A&P, what do antigens trigger? An immune response, right? And very often our example of immune response is antibody production, right? So you guys, if we get invaded by a bacterium with a certain type of flagellar protein, we'll produce antibodies against that very specific flagellar protein, right? So who cares, right? Well, it turns out that we can make antibodies against all the different flagellar proteins, all the different flagellar antigens, and then use those antibodies to identify specific strains of bacteria, right? And that process, you guys, where we use antibodies to identify specific strains of bacteria, it's called serotyping, right? So an example is, let's say that we have an E. coli, right? So you guys, there's so many different strains of E. coli, many different strains of E. coli. The ones living in your intestine, they're not harmful, right? They don't carry any toxin genes, right? But there are other strains of E. coli, folks, that have acquired toxin genes and can be quite deadly. So it's important if we, for example, have an outbreak of, say, a really serious diarrheal disease, we find out it's caused by E. coli, we need to know which serotype it is, right? So we can track back, where did it come from? How many people are infected? And again, you guys, historically, antibodies could be used to do that. So folks, the example of an E. coli serotype we want to use, it's right down at the very bottom, or actually about halfway through page 2, you guys. It's called E. coli serotype 0157H7. And for the lab exam, it's fair to ask you guys, what is that? It says 0157H7. I want you to know that the H is the flagellar antigen, right? And we can see there, folks, that amongst E. coli, there must be at least 7 different flagellar antigens, right? And this particular toxin-producing strain of E. coli has the 7th flagellar antigen. The O, folks, this is a little bit harder to understand, but the O is for the polysaccharide of lipopolysaccharide in the gram-negative bacteria cell wall. Now, polysaccharides they're not wonderful antigens, but you can get some antibodies against them. So antibodies against the O antigen, the polysaccharide of lipopolysaccharide, can also be used in serotyping to identify specific strains of bacteria, or E. coli in this case. So you guys, why do we worry about E. coli 0157H7? We're going to be talking more about it in the medical micro lectures, but I just want to give you a hint of why we're worried. We've got this serotype of E. coli here in California. Transmission is fecal orally, so wherever feces goes, if it's in it, if it contaminates food or drinking water, and if we swallow the food or drinking water, we're going to get infected. And the reason it's so serious, you guys, is it makes a really powerful toxin called the Shiga toxin. Okay. So we really, really worry about Shiga toxin production, especially in our children, right? So let's say our children swallow an E. coli 0157H7, it starts growing in our children's intestinal tract. The Shiga toxin, you guys, can be absorbed into blood vessels, and it causes blood vessel damage. Specifically, it shuts down ADS ribosomes, right? So we get absorption into the blood vessels, we have damage to blood vessels, we can get hemorrhage, so you can end up with a bloody diarrhea, and it's going to be frank red blood, you guys. It's not digested blood, it's the blood will appear in the feces. And to me, you guys, I would consider that as a parent a medical emergency. I would take my kids to urgent care, ER, call the doctor, because that could be a sign that the child is colonized with an E. coli 0157H7, right? So we have blood vessel damage, we have a bloody diarrhea. We can, because of the damage to the blood vessels, that can cause red blood cells to lyse. So we end up with a hemolytic anemia. And furthermore, if the toxin circulates to the kidneys, remember you guys from A&P, the kidneys are chock full of all these delicate little blood vessels, all these little capillaries. It can damage the kidneys and cause kidney failure. So in really serious cases, we have multiple problems going wrong, and when that happens, we call it a syndrome. So E. coli O157H7 and making Shiga toxin, it can cause the syndrome known as hemolytic uremic syndrome. Hemolytic describing blood cell damage, uremic describing an increase in blood urea. That's a signal that maybe the kidneys aren't working very well. Hemolytic uremic syndrome because multiple things are happening. And you guys, this is heartbreaking. This can be deadly in children. It can kill children, right? And again, we'll come back to it more in our medical micro lectures, right? But this is a bad serotype of E. coli. Good, okay. And then let me see what else here. All right, folks, and then the next thing we want to ask ourselves, well, how might we determine whether our bacteria are motile or not? And one is we can make a wet mount, look at it under the microscope, and just look for bacteria that are doing a series of runs and tumbles. That's one way to do it, right, just directly. But there's another way, you guys, we can do this indirectly, and that's by using motility agar deeps. And the recipe, folks, for motility agar deeps is on page 3. And what I'd like you to see there is that there's a really low concentration of agar. There's a very low concentration of agar. So you guys, this recipe is for 1,000 mils a liter, and you can see you'd add, for 1,000 mils, you'd add 4 grams of agar. Okay. Normally, for normal agar, like in our agar plates, or say our agar slants or agar deeps, per liter we'd add 15 grams per liter, right? So this is really, really low agar, and the reason is we want there to be so little agar so that the medium is solid, but that there's really low agar so our bacteria literally can swim between the agar molecules. We want them to be able to swim into the agar itself. Okay. So the way this works, you guys, is we have the motility agar deeps is what we call them. Right? After they're autoclaved and the agar is still liquid, the tubes are permitted to solidify in an upright position so we get these nice, great big deeps. And what we do to inoculate them, we use our inoculating needle. And what we do, folks, is we stab our bacteria straight down and straight back up. Okay. Now, we're not going to do this today as individuals, folks. We did this as demos already. So let me go grab our demo tube so you can see the results. All right. Now, to me, what makes this even more fun is that there's an indicator, and folks, you do not have to memorize the indicator, but it's here on your formula. It's called tetraphenyl tetrazolium chloride. And what's so fun is that if you have bacteria growing in the medium, it will turn that indicator red. So it lets you, it makes it easier for you to see after incubation where the bacteria are growing. So if we had bacteria that were non-motile, folks, we would suspect the bacteria should only be growing along the stab line, right? Because they can't move away from the stab line. They're just stuck there. So we'd think we'd only have red along the stab line after incubation. But if our bacteria are motile, right, they start growing along the stab line, they're using up all the nutrients, they're producing metabolic waste products. If you had a flagellum, would you want to move away from the stab line? Yeah, right, because move into new agar where there's more food and less metabolic waste product, right? So what's so fun, you guys, if your bacterium is motile, after incubation, you'll see red spreading out from the stab line. And this is one of those things, you guys, actually, it helps actually to come up closer and look at it, because I can see diffuse pink moving into the agar, and that's telling me my bacteria are motile. They're swimming into the agar, right, trying to find more nutrients, trying to get away from the metabolic waste products. Okay, so folks, what we would do is on Canvas, we'll put some photographs of really nice motility agar deeps, non-motile, motile, and that's also how we would ask you on the lab exam. We'd have really nice photographs, so it's really apparent, you know, these guys are only growing along the stab line. This other tube, I can see that the bacteria are moving into the agar, right? So bacteria moving into the agar, that is evidence that they are motile, okay? No growth into the agar, that's evidence that the bacteria are non-motile. Okay, you guys all right with that? Okay, good enough. Alrighty then. All right, folks, so now we're going to move to the 2 exercises where you're actually going to do something. And both these exercises, you guys, are pretty quick. So now we're on page 3 down at the bottom where it says Chapter 11 or Unit 11, Aseptic Technique and Transfer. So another term, you guys, for aseptic technique is sterile technique. So these are steps we take to prevent contamination with unwanted environmental microbes. So folks, what about if you were doing surgery, would you want to make sure that the surgical instruments were sterile? Yeah, because why? Yeah, you don't want the contaminated instruments to actually cause an infection in your patient, right? So that's pretty straightforward. We kind of get that concept. But folks, in the microbiology lab, aseptic or sterile techniques are really important because if we're trying to identify unknown bacteria, maybe a pathogen from our patients, we have to have a pure culture, just that pathogen. And we can't contaminate the culture with airborne microbes or microbes from our skin or microbes from inanimate objects. So in a microlab, we're always transferring our microbes from one place to another. Maybe to give them new food, right? Or maybe to do a metabolic test so we can identify them, or maybe to do drug sensitivity testing, which we'll be doing next week, right? So you already know how to do this. When we were doing our smears and gram stains, you guys were already practicing aseptic technique. But we're gonna do a different kind of transfer today, folks, so that we can continue to practice and develop our aseptic technique. So folks, the transfer we're gonna do today, I'm gonna walk you through it, folks, but on page 4, there's the actual protocol. So I'll demonstrate it for you, but if you're like, wait, that went too fast, there's actually step-by-step on page 4 for you for the aseptic transfer. Okay, and I'm always so bad at this, you guys. I should be a good role model and have my full PPE on, right? Because we're working with living microbes. So I should have my lab shoes, my lab coat, and I should have my gloves, okay? And for this, this transfer, you guys, what we're gonna do, we're gonna move E. coli, pure cultures of E. coli that Val made for us. We're gonna move our E. coli from an agar plate. Maybe they've been here for a while and we're worried they're running out of food, right? We're gonna move them to what we call a big butt agar slant. I don't know why that name just makes me smile. Okay, so you guys, a big butt agar slant compared to a little butt agar slant, what we do is we add a lot more agar, right? And when the agar solidifies at a little bit of an angle, you can see we've got a big butt, a big deep butt here. I can't even say that, right? Okay. And the reason we want big butt slants is we want to see if our microbes can grow in the anaerobic butt, right? The butt's going to be anaerobic. And then you guys, we're also going to see if they can grow on the aerobic slant. Now, how are we going to get our bacteria, you guys, in the anaerobic butt? Are you going to use your loop? No. I've tried it before, you guys. I'm so stubborn. If you try to use your loop and you try to stab the agar, the loop's going to go like that. Okay? So what are you going to use? Needle. The needle, right? You're going to use your inoculating needle. And let me give you the overview, folks. What we're going to do is we're going to do what's called a stab and streak. So we're going to harvest our bacteria, and we'll walk through this. And we're first, you guys, going to stab the agar, and we're going to try to go almost to the bottom. If possible, you guys, we don't want to touch the glass bottom. And the reason is, if your bacteria start growing in the butt, between the glass and the agar, if they start making gas, the gas will move up between the agar and the side of the tube and create an alternative way for air to get down to the bottom. Okay? But you guys remember, if you hit the glass bottom, it's not a big deal. We're just practicing, that's all, right? We're just practicing, so don't fret, don't fret. Okay, so you guys, what we're going to do then, we're going to stab the butt, and then we will withdraw partially, and then, folks, we're going to streak the surface of the slant, and let me show you how we do that. So historically, we streak the surface of the slant almost like we're a snake slithering along, kind of in an S-shaped motion. Okay, so let me do it over here, you guys. So historically what we do is we start down at the bottom of the slant, and as we're pulling our needle up, we're moving it in like a snake-like motion and an S-shaped motion. Okay? All right. So you guys, that's the overview of a stab and streak. And again, the function is you're trying to see if your microbes can grow anaerobically in the butt and aerobically on the slant. So you guys, let's just walk through the steps here really quickly. So let me talk you through the steps, and then I'll stop the movie and I'll give you the TSA slant so you can work on it, and then I'll start the movie again. Okay? So you guys, the way we're going to do this then, we have enough E. coli plates for 1 plate per pair. Okay, and you guys, please bring them back up in front here because all the other lab sections have to use them. So this is your source of pure culture. Okay, and then you guys, I'll bring around your big butt slant. We want to make sure we label it. So what would be your label? E. coli, good. Your name, right? Date and lab section, good. And where does it go? On the glass body. Good job, you guys. So you want to make sure that your tube is labeled. You're going to need your needle. And so is this sterile right now, folks? No, it's not sterile. So how are we going to sterilize it? In the Bacterial incinerator, right? And folks, do remember, only handle your needle or loop by the end here, because this portion, the metal portion, the handle will get really hot and will burn you. And furthermore, folks, remember, we've learned only insert like half the wire into the hot barrel. If we insert the whole thing, the junction between the metal handle and the wire will have metal fatigue, and eventually your needle will just fall off. Okay, so we're going to insert, and for how long? Only 5 seconds, right? And even if we had disease-causing prions, you guys, that should totally incinerate them, right? So, do I want to set my needle down after I've sterilized it? No, you don't want to touch anything. If you do, no big deal, just re-sterilize it. But you guys remember, often your test tube racks can act as a needle or loop holder. So I like to put my sterile loop or holder or needle there. And then, folks, we're going to take our pure culture of E. coli. Do I want to take the lids all the way off? Why not? Airborne, right? And oh my gosh, you guys, your plates will count the colonies. You take a look at your plates. It is so terrifying what we are breathing in, right? But anyway, the air is full of airborne microbes, right? So you guys, we always open the plate like a little clamshell with the hopes that the lid will prevent a lot of airborne microbes falling on the agar. Okay. And then, folks, if I had just sterilized my needle, do I instantly want to touch the E. coli? No, why not? We could fry them, right? We could fry our babies. So what we do is we just touch the agar where the E. coli aren't growing. If the needle or loop hisses and spits, it's too hot. Hold it there. When it stops hissing, it's cool enough. And then, folks, we're just going to take the tip of our needle and just touch that opaque ribbon where all the E. coli are growing. If you touch it, you're going to get millions of bacteria. And then we want to close the plate, right? Invert it. And then, folks, to do the transfer, what we're going to do, now in the old days we'd have a Bunsen burner flame and we'd flame the cap, we'd open it, flame the lips, but now we're using the Bacterial incinerators. So what I'm going to have you do, folks, we're just going to try to create some warm air currents that will hopefully push airborne microbes out of the opening of the tube. So the way we're going to do this, we're going to hold the lid to the opening of the Bacterial incinerator. Don't insert it, but just at the opening for a second, just quick, right? 1 second. I'm gonna take off the cap, you guys, and hold it with my pinky. I don't want to set it down. I don't want it to get contaminated, right? And then 1 second, you guys, warm the lips. And again, we're just trying to create some warm air currents to push the airborne microbes out, right? Now, remember, you guys, I've got my E. coli on my needle now, and this is always a hard part for me. You want to try to go straight down the tube. You guys, if I hit the outside of the tube, what has happened? I've contaminated it, right? And probably what we do is you say, okay, just put everything down, we'll spray the tube, we'll have you start all over again, right? So take some time to try to make sure that you're going to go right through the opening of the tube. And what you want to do, folks, again, you want to go straight down, almost to the bottom without hitting the bottom, withdraw, and then do what? Then you're just going to streak the slant, right? Like a snake. Good. Oh, yeah, that's great. And now what do I do? Just heat gently 1 second. Put the cap on. Heat gently 1 second. Do I want a tight cap? Always loose, right, to let it vent. Don't let it fall off, though. Just barely loose to vent. And really important, you guys, it's easy to forget. What's the last step? Yeah, always, if in doubt, you guys sterilize. If in doubt, wash your hands and sterilize. Okay? And then, folks, When you're finished with that transfer, we don't have enough room in our incubators for all the test tube racks. So what we're going to do, you'll find cans under the sink in the back of the room. Grab a can, try to decorate it, try to make it uniquely yours so somebody doesn't grab yours by accident out of the incubator. It probably wouldn't hurt to put maybe your team number, lab section here. And what we'll do, folks, is you can put 2, 3, or 4 of the tubes into the can. And again, guys, just check to make sure, is that cap barely loose just to vent the gases. And then it's going to go into 30°C incubator on, I think you guys were the 3rd shelf down on the right. It's got a Tuesday, Thursday, 1 p.m. Right, so we'll put those in there. Okay, so folks, what I'm going to do right now, I'm going to stop the movie and I'm going to bring your big butt TSA slants around. And then, while you guys are working, I'm going to be preparing our urine. Because our last exercise, you guys, is to try to isolate bacteria in what we call isolated colonies using streak plates. So we're going to learn how to streak a plate to get isolated colonies. And we're going to pretend that you're working with urine samples from 2 elderly women, and you suspect they might have bacterial cystitis, a bacterial bladder infection. So we're going to be working with, it's not real urine, you guys. We're working with pretend urine. We're going to streak out a plate to see if we can get isolated bacterial colonies and figure out if indeed these poor women have a bacterial bladder infection. Okay, so I'll stop the movie, you guys, and then I'll bring your big butt TSA slants around. One person per pair, come and get an E. coli plate. Bring them up when you're finished. Make your labels, and then you can start your transfers, okay?